Thank you very much, Michel. I think the microphone is on now, right? Yeah, you mentioned Templeton Prize. You didn't mention the National Medal of Science, which I got from President Bush, and a few other things that I have gotten along the way. Anyway, it's a pleasure to be here. As John told you earlier today, we have been organizing this colloquia dedicated to evolution from various points of view. And one of the reasons why, even before John came to UC Irvine, many years ago, I started to have symposia dedicated to evolution, had to do with my realization that, shocking as it would be to most of us, I became aware that most people in the National Academy of Sciences didn't know there was a science of evolution. They were quite willing to accept that organisms evolve, but didn't realize there was anything to study. And the best testimony that I have of that state of affairs happened in 1980, when the state of Arkansas passed a law that said whenever evolution is taught, equal time has to be given to the teaching of creation science. What had happened is, in the late 60s, in 1967, the Supreme Court of the United States have made a decision that states could not prohibit the teaching of evolution. So the way they wanted to get about it was originated by an engineer from Texas who sent a project of law to 15 states, and it was introduced in 15 states, which required that whenever evolution is taught, evolution science, then you have to dedicate equal time to creation science. And of course, there was no creation science, and the point was that people confronted with that law, they were going to stop teaching evolution, which is what they wanted. Well, at the time, it's 80 years ago, it's 35 years ago, I was already a member of the National Academy of Sciences, and I was also at the time president of the Society for the Study of Evolution. So I went to see the president of the National Academy of Sciences, Frank Press, who had been the science advisor to President Carter and was a very distinguished scientist. And I told him, we had met for lunch, that I thought that the National Academy of Sciences should take a position on this matter. And evolution? Huh? Oh, so you look at me surprised. So I said, yes, we have actually a section of the Academy dedicated to evolution. I was evolution, ecology, and population genetics. Now these sections have been split into two. So he went to the book, and there were a grand total of 18 members out of the 1,800 members of the Academy. He said, why should the Academy be involved in something that affects so few people? And I said, Frank, this is not a question of how many people are affected. This is about the survival of rationality in our country. If we teach, some statements of Genesis as if they were science, which is what the creation science presumably implied, where are we going to go? So eventually he appointed a committee, and the Academy got involved at that time, and a later trial that happened in Pennsylvania, and things are reasonably well. But as I said, it was shortly after that meeting with Frank Press in 1980 or 81 when I decided that we need to increase the visibility of evolution in the Academy and in science in general. And I organized, together with some other colleagues of mine in Irvine, 4 symposia, one every 2 years, to celebrate the anniversaries of the great books that develop what we can call the modern theory of evolution. The 1936 book of Dobzhansky, Genetics and the Origin of Species, then the book by Simpson about paleontology, a book by Meyer about animal species and evolution, zoology, and the book by Stebbins on animal, plants, and evolution. So every 2 years, we were organizing one of these colloquia. Then 4 more years go by, and then my dear friend and former student John Avis comes to Irvine, and now knowing John and that he's a man of energy and creativity and willing to organize things, we decided to start this new series in the light of evolution, which is, as he has pointed out, and as you know, this is number 9. The next one next year will be number 10, and then we'll take a break. But by now, everybody in the National Academy of Sciences is aware that evolution exists as a discipline. I should point out that the National Academy of Sciences sponsors 2 or 3 colloquia like this per year. For the last 9 years, one of the 3, or the 2 or 3, has been on evolution. So as I said, our visibility is quite substantial. Anyway, it's nice to have you here and to provide this lecture. I am going to say things which are very obvious. Some of them are going to be related to what Carol said earlier. And if you have had enough wine and want to take a nap, I will not complain. So one of the contexts within which this issue of whether humans should be cloned, one of the contexts in which it arises was the claim that became surprisingly fairly popular in the 1950s and 60s and 70s, that natural selection was not working any longer on humans, and therefore that our genetic makeup was going to deteriorate, and therefore something should be done about it, and cloning and other things that I will introduce later was being proposed. So the question that I am arising here, evolution in modern humans is indeed the case that natural selection has stopped, and the point I am going to make obviously is not. But I'm going to first start with 2 cases of those that can be cured, genetic diseases. The notion was particularly introduced by H.G. Muller, the second geneticist to obtain the Nobel Prize in 1946, who used to say, in a sentence that I will quote again later, the more disease we cure, the more we will have to cure in the future. Because if those who are sick, hereditary diseases, survive and reproduce, then the frequency of the genes that cause the diseases will keep increasing. So let us explore that for a moment, because H. Muller was a great geneticist. He got the Nobel Prize for having discovered that X-rays induce mutations. The first geneticist to get the Nobel Prize was Morgan, but then a few years later, Muller got it. So let us consider one disease, phenylketonuria, which is, we know very well, the genetic causation, is an allele, the PKU allele. The disease consists in inability to metabolize phenylalanine. So the amino acid accumulates and causes considerable harm, severe mental deficiency, neurological disorders, and early death. The incidence of phenylketonuria is not trivial, 700,000 worldwide. The allele frequency is 1%. So 70 million in the world are carriers. If the disease is diagnosed very early, I mean by very early, just less than 2 years of age, and the child is provided with a diet free of phenylalanine, and there are some substitutes that can be added in the diet, the child can survive and even reproduce, go beyond the reproductive age. Now let us consider a dominant mutation. This is retinoblastoma RB1. There are several kinds of retinoblastoma. This is the most common and the most severe. It's a cancer of the retina, which appears first in one eye. It spreads to the other eye and the cancer goes to the brain, and the child dies, usually very, very early. The incidence is about 1 case for 30,000 live birds. So the allelic frequency, the gene frequency is about 4 in a million. Now this also can be cured. It implies enucleating at least one eye, very often both, and then the child can survive. As you can imagine, it's not the most wonderful kind of life that this person can have. It can survive even to reproductive age and reproduce. So here is what Muller thought was going to be happening a few hundred years from now. In fact, in some cases, in his writings, he speaks of 300 to 400 years from now. And what we will have is a situation where the amount of genetically caused impairment suffered by the average individual, even though he has all the techniques of civilization working to mitigate it, must by that time have grown. People's time and energy at that time would be devoted chiefly to the effort to live carefully, to spare and to prop up their own feebleness, to soothe their inner disharmonies, and in general to doctor themselves as effectively as possible. Next statement, everyone will be an invalid. Our descendants' natural biological organization would in fact have disintegrated and have been replaced by complete disorder. In fact, he says it would be in the end easier to factor a human de novo, as he put it, to create a new human, rather than trying to cure the people who already had such miserable lives due to their deteriorating genetic makeup. And this is the kind of a statement that he kept repeating. The more sick people we now cure and allow them to reproduce, the more there will be cured in the future. Well, is that true? It has to be true up to a point. But Muller was, whom I happened to know him in the later years of his life, happened to be a great geneticist and a great human being, but he was not a population geneticist. He did not understand how populations work. By the way, this is the way he anticipated humans would look a few hundred years from now. Artificial members, artificial organs, and all of that miserable. So take the PKU allele, frequency of 1%. Assume for a moment, which of course is not possible, every person of these many thousands in the world who suffer from PKU were to be cured from very early childhood, protected, and would reproduce as much. Everybody suffering from phenylketonuria will reproduce as much as the average person in the population. Well, there's a very simple equation in population genetics that tells us how long it will take for the gene to duplicate in frequency, namely 1/Q. The frequency of the allele is 1%, 1Q is equal 100. So it will take curing everybody and everybody reproducing that had this disease, it will take 2,500 years or 3,000 years for the frequency of the allele to double. Now, no matter that if the frequency of the allele increases even very, very little from one generation to another, as it would be the case, still there is a problem. But the problem is not the kind of problem that Muller envisioned. And certainly I fail to be worried very much about what the situation will be 2,500 years from now, when the frequency of the disease will have increased by a factor of 4, the frequency of the allele by a factor of 2. I mean, science is what genetics is a little over 100 years old, and barely. Molecular biology, half a century. You can imagine what would happen in a few hundred years, not to say 2,500 years. And I will come at the end to some of these possible things that could be done in the future. But obviously mankind will be able to do things that would avoid the implications of this deterioration of the genetic makeup. Well, genetic diseases that now can be diagnosed. Here I put a few examples of them. There are actually several thousand of them, close to 2,000 diseases that have a genetic cause. I'm going to call your attention to two of them, of this example, of this sample, sickle cell anemia and Marfan syndrome, because I'm going to refer to the two of them for very different reasons later. So what if, rather than consider just one disease like retinoblastoma or one disease like phenylketonuria, we take all the diseases that presume to be about 2,000 that have genetic causation. They affect about 2% of the world population, about 150 million people. There's a lot of people. And again, little increase in frequency will increase substantially the number of people who have to be taken care of. Now, these hereditary diseases, if we take into account mental impairments, those seem to affect about 3% of the population of 225 million people. Some are hereditary. Here we have no much idea, I don't think anybody does, of precise idea of how many of them are genetically caused or what percentage of the mentally impaired people are so for genetic reasons. Well, now I want to take into account the possibility of practicing genetic therapy. In the case of phenylketonuria or in the case of retinoblastoma, these are the traditional way of doing medicine, protecting, or having surgery and the like. But now we know that we can correct the genetic makeup of individuals. And one of the early examples have to do with sickle cell disease, as it is called now. It used to be called sickle cell anemia, which is very common, particularly in the tropics and particularly in sub-Saharan Africa. The cause of sickle cell anemia is because of replacement of valine for glutamic acid in the 6th position of the beta chain of hemoglobin. In our adult hemoglobin, so-called A hemoglobin, which is about 98%, the A hemoglobin is 98% of the adult hemoglobin, is made up of each molecule of hemoglobin is 4 chains, 2 alpha and 2 beta. So if the beta chain has this mutation, the hemoglobin is not able to associate properly with oxygen. The red blood cells acquire a sickle shape, like, and they don't function, and eventually cause death. So it's a severe disease which starts to manifest itself in the early adolescence and usually causes death before maturity, before 20 years of age or so. Now, this is a description of it. So this is sickle cell disease. It's due to homozygosis for the sickle cell allele, that is SS, which is lethal before reproductive age, about 275,000 births worldwide per year, not a trivial amount. Sickle cell trait is due to heterozygosis for the allele, so the genotype is AS, and the normal genotype will be AA. Now, the people who suffer who are carriers of the sickle cell gene have virtually no symptoms. Occasionally, some abnormalities happen in some individuals, but by and large, 99% of these people are, by all means, normal people. The SCT, the sickle cell trait, the carriers of the gene, but is prevalence in sub-Saharan Africa is 15% of the population, where 80% of the worldwide malaria occurs. The reason to bring malaria into account, because the explanation which was suggested a long while ago of why the sickle cell gene has high frequency is because in heterozygous condition it protects against malaria. And of course, malaria is the largest cause of mortality in the world. I was very amused these past days, not that the matter is not serious, when they were saying Ebola has already killed, I believe it's 9,000 people since it came, this epidemic. Malaria kills about 500,000 children a year. So compared with that, Ebola is nothing. Of course, we are now investing a lot of money, this country and many foundations and the like, trying to deal with malaria. The suggestion, based on various calculations of correspondence, that sickle cell anemia protects against malaria has been persistent, and I'm going to give you the most recent results, and actually the most interesting results which show the association. This is an experiment study done in Gabon by scientists from Montpellier, from the institution with which Michel was associated with many, many years, some of whom work in Gabon. So they study Gabon because it's one of the places where the incidence of sickle cell disease is highest. And so they took samples from all the provinces of Gabon, in fact, from almost all the villages. And what you can see just in this map, which is sort of impressionistic, not detailed, that you have here the yellow, the areas where the prevalence of the sickle cell trait, this is the heterozygous, is less than 10%, and here is the red where it is higher percent. And then when you look at malaria, you can see that where the malaria is lower, SCT is lower, where it is higher than 30%, the SCT prevalence is higher. So there's this correlation, and they did a lot of statistics. This is now under review for publication in the Proceedings of the National Academy of Sciences. It's not yet published, but there are a number of analyses they have done which are very persuasive. So again, to summarize the experiment, they sample about 4,000 individuals coming from all 195 villages over the entire republic. 859 had the heterozygous genotype, 3,100 had the homozygous normal. They don't find in their samples, which were individuals of 15 years of age or more, they don't find a single one who will be really suffering from sickle cell anemia. All have died before that age. So what they calculate is that any increase in malaria prevalence from 40 to 50%, an increase by 25%, is associated with an increase in the sickle cell trait of from 21 to 22%, or about 4.3%. And in other ways, as well as this one, they sort of provide strong evidence that really this is the sickle cell trait that is protecting them against malaria. The incidence of the HBS allele in Gabon is about 15%. In the United States, it's almost 1%. And of course, the incidence of the people suffering from the disease is about 1 in 10,000. Almost all of these are among African Americans, people who brought the trait from from Africa and has become here, eventually becomes expressed due to homozygosis and the like. Now, what can we do at genetic level to cure the sickle cell disease? Well, actually, it turns out quite a lot. The technique, this is related to some of the things that we were told just before dinner time. The technique in this case consists of extracting stem cells from the bone marrow, essentially, where we have these stem cells that produce the blood cells. We replace our blood cells, about twice a year on the average or something like that. Our red blood cells are dying all the time, and we are producing new ones. So you get cells from the spinal cord. Then genetically, one can introduce the allele which is missing in the beta chain using a plasmid. And then these cells, these stem cells, are put back in the spinal cord. And now the individual produces still many red blood cells which are defective and will take the sickle shape, but produces enough normal red blood cells to survive fairly well and to adulthood. So that's one example of something that can be done genetically to cure diseases. The other example that I'm going to use is the severe combined immunodeficiency, SID, as it's sometimes called. This is a much more severe condition than almost anything you can imagine. These are children who have the whole immune system turned off because of problems of control, gene control, and therefore they have no immune defenses whatsoever. The first time that these children get exposed to some bacteria in the air or some other malignant microbe, they get infected, they die. So the only way they can survive is by, they are called bubble children. They have to survive by being enclosed in a glass bubble, literally, where all the air that comes in is filtered. Well, it's a miserable life, miserable condition, very expensive, and in any case, they rarely survive more than a few years, 2 or 3 or 4 at the most. Well, the thought was, again with the advent in the towards the end of the last century, at the beginning of the genetic manipulations that we could do, genetic engineering, that to introduce a plasmid that will allow these genes, the immune system, to be turned on so that the child will have the defenses. This was started in 3 hospitals. The most dramatic one and the example, the one I'm going to use is from the hospital for sick children in Paris. They first did it with one child. The child was cured. They tried another one, was cured, and another one, and then started to do 6 or 7 more. Then by the time they were curing the 11th or 12th one, suddenly this child, one of the children, developed leukemia. And it occurred to them, as they are saying in this press conference, that probably the leukemia was caused by the genetic therapy. And actually what happened is within a short time after this press conference, several of the other children have also developed leukemia. So they stopped the treatment, and then about 2 years later, by the way, the 2 hospitals in the United States that were doing it didn't have this problem, but they could have had it. Anyway, after 2 years later, they have restarted the treatment again. They thought that they have a new way of doing it, and it has been a mixed success. They have some occasional cases of leukemia, but rarely now, and they just cure these children. here you have an example of how these children are, they live when they suffer from this terrible disease. Well, gene therapy trials, now this is relatively old slide, but things have not changed very much. There are lots of countries now which are carrying on lots of gene therapy trials in the United States, in Europe, great concentration in places like Switzerland, there, and others. But anyway, there are many, many diseases for which gene therapy is being tried, and some are becoming, the situation is being solved, so the therapy is discovered that cures. But, you are curing the disease. You are not changing the genes, the genes that are going to be inherited. Even as you cure the genes that the individual carries that are causing the disease, like in the example that I was giving with sickle cell anemia, still your gene is going to be present in the sex cells and is going to be transmitted to the following generation. Now, this is examples of, again, this is clinical trials for genetic therapy, and here is a seed, and also, well, anyway, some of the other ones that could be mentioned. So we come back to H.G. Muller's pessimism, because still the case is that the disease is cured, but then the person is going to transmit the gene to the following generation if the person is able to reproduce, and then you have the issue of having to cure the people in the next generation. So again, Muller's pessimism, the more sick people we now cure and allow them to reproduce, the more there will be to cure in the future. And here we come into the Brave New World, and much of it pushed in his day by Muller, but also by others. One is the possibility of genetic engineering of reproductive cells. Will that be possible in the future? Along this line, I don't know there has been any progress, that is, to correct the genes in the sperm or in the eggs. But there are other proposals pushed by Muller. One was germinal selection, which I will explain what he meant in a moment by those terms, and then human cloning. Well, by germinal selection, what Muller said is, assume that we create banks where very distinguished men can deposit their sperm. And then people marry, and the women will have the option of being fertilized, not by the sperm of their husband, but by sperm taken from the bank from a very distinguished man. And he says, no woman will refuse to have a child fathered by the likes of, what happened here? Of Leonardo, Lincoln, or Lenin. He mentions in different papers different names. I put these three here for two reasons. One is for amusement, because all are L's, the first name. Second, as I don't know whether you are familiar with Leonardo da Vinci, one of the great geniuses in the history of mankind, but a pedophile who went several times to jail for abuse of children. And if you remember the Last Supper painting, the youngest of the apostles, St. John, who is resting on the shoulder of Jesus, it was his lover at the time. It's a portrait of his lover. Well, I'm laughing. It's not necessarily a matter to laugh, but I would imagine many women would not want to have a child fathered by this, pedophile, genius as he was. Well, Lincoln, as now we know, I think it's virtually certain, suffered from Marfan syndrome. So if he would not have been assassinated, his condition was such that the experts say that he would have died probably in 2 or 3 years, just from Marfan syndrome. So again, he's proposing somebody whose sperm will be used by all sorts of women because the genetic endowment of mankind is deteriorating. It's a person who carries a severe disease. And with respect to Lenin, this is from one of his early articles. Muller became a communist, in fact, immigrated to Russia, became very disappointed very quickly, and got out of Russia by the time when he thought it might not have been easy, with the excuse of coming to help out the Spaniards in the Spanish Civil War in the 30s. And of course, he changed his politics completely. At the time I knew him in the middle 60s, he was politically very conservative. So in his later lists, Lenin is not listed as one man who anybody want to have his sperm for in order to have a child. Well, the point that is implied here is not that these men will not be desirable men, but first of all, you have a bank of sperm from men who have proven to be very distinguished and eminent. You know, they are no young fellows anymore, and the sperm deteriorates with age, as we all know. So, I mean, this is not the best way to improve the genetic makeup of humankind. He says, eventually, at the time, he thought that the technology was not available. One could do egg flushing from women's ovaries, and as the eggs would mature, and create also banks of eggs, of human eggs, and then a couple that would marry will select the mother genetically and the genetical father and have fertilization in vitro, and then it's implanted in the mother, and that's how he envisions that the genetic makeup of humankind will be protected and improved. Well, this seems so almost ridiculous, but some people took it very seriously. William Shockley, Stanford professor and industrialist, who is one of the co-inventors of the transistor and got the Nobel Prize in 1956, invested a lot of money in creating clinics where men, distinguished men, could deposit their sperm. He actually, the rules were they were men who had won the Nobel Prize, just like he had. But I think eventually that was relaxed, and it was some men that would be accepted as being very distinguished. I am not aware, although I have looked into it, that in any of the clinics any man ever deposited the sperm, except perhaps his own. But eventually they were closed for lack of use. The last one in the year 2000. Well, so other possibilities, cloning, and we have heard enough, and you all know enough about the difference between cloning genes, something which is done by the billions in any one of our genetic laboratories every day, or cloning plants. You know, Michel pointed out that I am a wine grape grower, and not only I'm a wine grape grower, I'm a wine grape dresser. You know, these are vines. And this has the name of the main wine-producing region in Spain, Rioja. They gave me a prize there, and they gave me the necktie. Anyway, as you know, plants are very easy to reproduce clonally. In fact, most fruits and many other kinds of plants the plants that we use in our diets are reproduced by cloning. Let me see. I am going to use 2 minutes to tell you a story, which I hope you find it amusing. I'm a serious wine grape grower, and I have introduced novelties at many levels in the wine grape industry, in the wine industry in California. And one of them, about 10 years ago, was to identify wine grape that probably most of you have not heard of, called Teroldago. And I thought that for the conditions in the area around Napa, the conditions of climate, the soils and the like, there will be a grape which is difficult to grow in many places that will do very well. So I planted 20 acres. Of course, no winery wanted to buy the grapes from me, so I did in this experiment what I did with others. I will give them 10 tons of grapes to this winery and 10 to the other winery and 10 to the other winery. And don't tell the winemaker what it is. Ask him to make the best wine possible and then try it. Well, people went crazy for this wine, Teroldego. So by vine takes about 5 years to produce the first full crop. For 4 years, you already have a crop. So a few years have passed since I have planted. I have gotten some of the plants from Europe, from Italy. So now a number of years have passed, and suddenly the wineries are going crazy about this wine. So now everybody wants to plant Teroldego, but no nursery, has plants, the cuttings to plant Teroldego grapes. So about 2 years ago, 3 years ago, whenever this first happened in large scale, and when they were all the people that produce the nurseries, that produce the plants that then the agriculturists plant, they all wanted to grow this little Teroldego. So as you know, a vine, I suppose you must have seen them, they produce canes which are tremendously long. Any one produces each vine 200 or so of these tremendously long canes. Then the frost comes, usually in the area that I'm talking about, Northern California, around mid-December or something like that. All the leaves fall and you have these canes, and then it's when the farmers will start to prune the plant. Well, I have these hundreds and hundreds and hundreds, 20 acres of vines, each one with hundreds of canes. So I sold them for each piece of cane like this for 50 cents to the people who have these nurseries. They were going to produce the plants for the following year to start to provide them to other farmers. So I made about several $100,000 just selling sticks of cane, from vines. So it's easy to reproduce vines clonally. That was the point. The story was, well, I hope for some amusement. It certainly was amusing to me. So now we have also the possibility of cloning domestic animals, and we heard just a couple of hours ago all what we need to know about it, or all what we want to know. And then what about the possibility of cloning humans? I want to make a few statements about this, because it amazes me that some people have in the past, and still some are speaking about the possibility of cloning humans in the future. Well, first about cloning animals, as Clara was telling us, you have all sorts of problems. Here you have a statement from Jan Wilmot, is the one who cloned the first mammal, the famous little dolly, and his survival rates, this is several years later, are still very low, less than 4%, as we heard earlier. And most pregnancy failures occur just before term, which would be devastating and cruel for humans. Imagine if most the pregnancies will go on for several months, and just very close to term is when the spontaneous abortion would take place. Now, since then, of course, have been successes in other labs, and we have been told about it. But the fact is that most of these clonings produce defective animals. Only a few survive. Again, we have been told about it. And even Dolly itself needed a special diet and had to be sacrificed eventually when still very young because of multiple organ failures and accelerated aging. Well, strange as it may seem, around the year 2000, there were several groups of medical practitioners over the world were very interested in cloning humans. And the two who got the most attention was these two guys, Severino Antinori in Rome, and Panos Savos, somebody of Yugoslavian origin in Kentucky. In both cases, particularly in the case of Severino Antinori, he claimed that he had over 2,000 women in whom he had implanted eggs with nuclear transfer. That is, had taken the nucleus, as we were explained, from somebody source, having from an adult cell, have put in an egg cell, and have been implanted. He got a lot of attention in the media and all of that. Nobody ever heard at the end what happened. I very much suspect that he was making these stories and was wishful thinking, so there is no reason to believe that he actually did all these 2,000 or so implantings that he claimed to have been doing. But I'm going to finish the claim that you actually can clone a human being. You can clone genes, but you cannot clone humans. You cannot clone, truly speaking, even animals or plants. With plants, yes, like with the vines. But look at this experiment made in two experimental stations owned by the Carnegie Institution of Washington. One of them is at Stanford, very near University of Stanford, 100 feet above sea level. Another one in Mather, this station where I have been, and I have been doing research there, is very close to the entrance to Yosemite, about 4,600 feet, and Timberline at the the highest altitude at which you can grow the trees or other plants will grow at 10,000 feet. So the experiment, this is an experiment done quite a few years ago, and this is one of the samples. They did it with many replicas. Is you take cuttings from plants, say, from Stanford, and then you plant them in Stanford, you plant it in Mather, and you plant it in Timberline. These three are clones. They're very well here, so in Mather, dies there. Then you take the clonings from Mather, these are clones, so they're cuttings from the same plant, and again, that's okay at Stanford, better in Mather, very poorly there, and the same for Timberline. So, which one of the three genotypes is best? Depends on the environment, and what environment is best depends on the genotype. The point is that you have this incredible interaction between environment and the genotype, and of course that's much more important for animals than for plants, and it's much more important for humans than for any other animal. Let me show you an example of animals. This is an experiment which consisted in taking rats, put them in a maze with food at the end so that the rats will try to get there, and then counting the mistakes, the errors that rats would make, and take the ones that would make fewer errors and breed them, and the ones who would make the most errors and breed them, and do this generation after generation. After a few more than 100 generations of breeding the smartest guys every generation, and also the stupidest rats every generation, they got genetic differences. When they did the test, what I call the normal environment, in the way they normally were doing their test, the bright rats did in the average less than 120 errors. The dull rats are close to 170. Almost a 50% difference. That will be like the difference in IQ between 80 and 120, something like that. It's quite a genetic difference. But what happens when you change the environment? They took the same rats, and now they ran them in a very restricted environment. They were crowded with very little food, very little water, and then the bright rats suddenly are making as many errors as the dull rats. And when they breed them in a very stimulating environment, then the dull rats, which were so stupid there, they become almost as smart, and in fact, statistically, were not significantly different from the bright rats. Well, I think that I'm reiterating something that is obvious to most of us, that you have this genotype-environment interaction, and that which genotype is best, as I was saying a moment ago, depends on the environment. Which environment is best depends on the genotype. Now, our environments are much, much more complex than those of the rats, not to speak of those of the cinquefoil, of the Potentilla glandulosa, by the way, is the popular name of the scientific name of that plant. The reason why they used it in this experiment is because it grows wild in California at all altitudes, from very near sea level to up to the tree line. Certainly our environments are much more complex than are those of the Potentilla glandulosa. Cloning is duplication of the genes, not duplication of the individual. The individual cannot be duplicated. And here I am going to quote another Nobel laureate, George Biddle, who got the Nobel Prize in the late 50s, in 58, I believe, and eventually became president of the University of Chicago, commenting on the proposals of Muller about, cloning people to improve our genetic makeup. He says, few of us, well, you have read it already, but I will do it. Few of us have advocated preferential multiplication of Hitler's genes, yet who can say that in a different cultural context, Hitler might not have been one of the truly great leaders of men, or that Einstein might not have been the political villain. That's obviously true. And he comments in other places where they make this kind of comment, that in order to, let us say, not to talk about Einstein, but I'm going to talk about myself, if I am pretentious enough to think that I have such a wonderful genome, and I am such a great individual that I should be cloned, in order to clone me, they will have not only to have my genes, but they have to have put that egg in my mother's womb, and my mother is not around, and I would have had, that new individual would have had to go to the same schools in Spain and emigrated to the United States, and on and on. The environments cannot be repeated. So if you try to clone an individual, you will clone the genes, but you would not necessarily get the same individual. And yet, as I said, there are some, according to the press, in some occasions, there have been some individuals that were trying to clone themselves because they thought they were so great and so good. And in fact, in the case of Muller, he was proposing that eventually that becomes a social program, so that the society starts to clone those individuals who are the best. And of course, there are legal prohibitions. As this conversation was going on at the beginning of the century, in the early 2000s, many governments in various countries made it illegal, even the attempt to clone a human individual. Here you have the Canadians allowed for stem cell research from embryos under specific conditions, but banning human cloning, and the same with the French Parliament and the Japanese and many others. So 2 words now about what I call here therapeutic cloning. So obviously reproductive cloning is not something that we'll ever want to do with humans. I don't see that now or 2,000 years from now. But therapeutic cloning, what I mean by that is something that again was referred to earlier. You can have nuclear transplant of, let's say, my genetic makeup, put it in an egg, and then you can develop that egg and in a way that it will produce a kidney or nerve tissue. Then, of course, you could do a transplant rather than having to borrow the kidney from somebody, from a donor, you could do it that way through this technique, which, as you may know, is now being explored in many places by a few pharmaceutical companies and a few laboratories. The possibility of using stem cells or even egg with a nuclear transplanted to try to develop specific organs or tissues. You can only imagine if we could be able to produce nerve tissue for the 100,000 people or so in the United States which are in wheelchairs because of car accidents, and they have a minor wound. It's very often very minor in some way, by some way of describing it, in the spinal cord. But because it's the spinal cord, they are now completely paralyzed. A wound that, if it would happen in our skin, will be cured in a few days. Now these people are paralyzed for life. I am no doctor and I don't know how these things could be done, but I can only dream that someday it will be possible to take care of these people through this kind of therapeutic cloning. So, organs for transplant, fine. Nerve cells for spinal cord injury, fine. Thank you very much.